The symptom, the discoloration of the seeds is mainly because of tomato spotted wilt virus disease. Then one of the important one is necrosis of terminal branches of the young leaves. Necrosis, that means we can see the dieback symptoms. The first, the necrosis we can see in the youngest leaves uh, at the growing tips, and the branches we can see the discolored streaks, and the affected plants may have one-sided growth habit or maybe entirely stunted and have drooping leaves, and it is almost looks like a wilting. Now, please try to see the difference between the physioremial disease, bacterial disease, and all these is common. And only thing is you have to observe the symptoms. Mainly, you can see the if it is plants or terminal branches, it has curling, and definitely we can say the such plants are affected with this tomato spotted disease. Then you also see the if you find disease, and also on the fruits, we observe any kind of concentric rings which is pale reddish or yellowish in color with a reddish background of the ripened fruits. So if you find all these symptoms, definitely we can say it is such plants are affected with this tomato spotted wilt viral diseases. This is a picture we have to see that the youngest leaves it is expressing the curling, then yellowing, and necrosis of the tissues. So this is a typical symptom of this tomato spotted wilt virus disease. So here we can see the fruit surface where we can see the presence of this concentric rings, which is yellowish in color, mostly with a reddish background. This is also one of the typical symptoms for the identification of this problem. No, but usually this tomato spotted wilt virus, which also going to cause the groundnut bud necrosis virus. This is in groundnut. And this is the leaf which is infected with the tosco virus, tomato spotted wilt virus disease. So these are the some of the pictures which is going to give some idea for this identification of the disease at the field level. So with respect to the etiology, this is the virus which is the negative single stranded RNA virus. It is a negative single stranded RNA virus, and it is belong to the family Bunnyviridae. So Bunnyviridae, and with the genus it is a Tospovirus. The Tospovirus means tomato spotted wilt virus disease. So most of these the Tospovirus viruses are transmitted by these the thrips. The thrips is the main vectors which are going to transmit the disease from one plant to the another plant. Since the virus itself, it will never going to move. from one place to the another place, it needs some kind of agent for movement, for their movement. So for that, the virus is going to make it less of this thrips as a carriers for spreading the disease from one place to the another place, or the one plant to the another plant. Here, this virus is a persistent virus. You know, this virus will be there inside the vector for many hours. It is a persistent virus, but it is not a transviral transmission. The transviral transmission means, so that means if it is the adult thrips, if it's going to give the second generation of the insect, we don't find any kind of virus particles in the vector body. Such virus, we can say it is a non-transviral in nature, but it is a persistent virus. Whichever insect which is going to take the virus particles into their body, the virus particles remain, it is going to viable in such vectors only. But their offsprings, we don't see any kind of virus particles. But it is not transmitted by through seeds. It is not seed-borne in nature, and also through contact, leaf contact. But only thing is the seed discoloration is very much problematic in this disease. And but it is and also transmitted through sap as well as through grafting. No graft. Then for with the local lesion host here, Nicotian and Petunia, they are the indicator host for this tomato spotted wilt virus. Nicotiana, you know, Nicotiana tobacco, Balsamina. There's the main indicator host for this diagnosis of this virus disease in tomato. And with respect to the mode of spread and survival, you know, the primary source of inoculum. So most of the viral disease, they are obligate parasites. That means it requires a living host for their survival. So for the secondary source of the secondary spread of the disease, mainly the Franklinus kuddelgi and the Cytothrips dorsalis. These two, the thrips, which are going to transmit the disease. Here, mainly the adult thrips can able to transmit the disease, not the young ones. Only when the larvae or the young ones acquire the virus from the infected plants, such adult thrips can able to transmit the disease. Since the adult thrips, which is having very good strong stylite, so with the help of the stylite, which it can be able to transmit the disease. But only thing is the nymphs is going to acquire the virus particles. For acquiring the virus particles, the nymphs will going to acquire the virus, but transmitting the disease. The important vital host for this type of virus disease, yeah, the then Chrysanthemum, Dahlia, Jarabera, and the pepper, potato, zinnia. So they are having very good host for this the main host. With respect to the management, so managing the insect is the most important. Important one is you have to take the seedlings under packet condition, especially the nylon. So the nylon is to drives the so having the 50 size, size mass, it will never going to permit the thrips to enter into your the seedlings. Use of this very important. And you can go for remove of the as well as the weed host. So and also we can use some the Crotalaria gentiana as a backdrop along the border. So to the movement of this, for that you go to grow this the green manure crops like crop, then we can go for use of sorghum or coconut leaf extract. So sorghum coconut leaf extract definitely going to give the better result on managing this virus disease. Then we can also go for the management of insects. You know, the insect for that use of the insecticide like imidacloprid, 0.5 ml per liter water. In market is available in the name of methoxystones. The Piperonil, it is at the rate of 0.1%. In the market is available in the name of Regent. You know, it's very good chemical, especially for the thrips management. Also use a rate of 1 kg active ingredient per hectare, or acephate at the rate of 1 ml or 1 gram per water. All these insecticides, especially the insecticides, which will go to take care of the pecking pest. Then One more important problem in tomato division with respect to the viral disease, tomato leaf curl virus disease. No, tomato leaf curl virus is a causal agent of disease in tomato. The disease appear in severe form, especially in September to November. Only this month, when there is the number of insect vectors. In our virus multiplication, it is only for this vector multiplication. More number of the vectors, more will be the disease. So here the main important symptoms of the disease is the growth. The common symptoms, most of the diseases, and the leaves, downward curling at the roll, uncurling and twisting and chlorosis. The main important symptoms in the leaves. Then leaves, especially the older leaves, it's leathery and brittle in texture. So because of the continuously these viruses take their nutrients for application. Application of the viruses, it's going to mainly it's going to depend on the host. So it has been the curling and twisting and stunted growth. Kind of symptoms in the plants. Then the flower drop is common in the plants which are infected with this disease. Thereby, you find if any flower drop, obviously we can see the dropping of this flower. Very few number of fruits in the plants. Then the disease plants which produce the partial any kind of fruits because of the severe dropping of flowers. You find kind of fruits which are very smaller in size and deformed, and it is not suitable for market. So some kind of symptoms of this tomato leaf curl virus disease. Now you can see the symptoms of the disease very small size leaves and the occurring kind of cup-like shape, and you can see the reduction in the internodal length, and you can see the very few number of flowers in the plants, and the curling of the leaves. Then here also we can see the like appearance of the leaves. The etiology, it is belong to the standard DNA viruses. No, tomato leaf curl virus disease. It is a single standard DNA virus, and it belongs to the family Geminidae and belongs to the genus of Begamovirus disease. Begamovirus disease. It is a propagative and circulative virus and transovarial transmission. In grapes, in this case, it is a transovarial in nature, propagative and circulative in nature. And that means in the second dimension, the viral virus vectors also we can see this the presence of particles. These are bipartite viruses, especially in northern India, where we can see the two genomes, DNA A and DNA B. That is tomato leaf curl virus. Tomato leaf curl virus is a bipartite virus. Only we can see the monoid and monoparticle. You know, only the DNA A will be there. But in northern India, we can see the both DNA A and DNA. And this it is abbreviated form here. It is T capital, O small, and remaining L, C, and D capital. Okay, it is a tomato leaf curl virus. It is a genuine virus. It is transmitted by white flies. You know, the white flies cause this. This is many cause. White that is the botanical name of this white fly, but it is seed-borne. It is not seed-borne in nature, but it is actually contaminated in nature. Not transmitted through sap, but it can transmit it through grafting. And the epidemiology of for this tomato New Delhi virus, you know, it is a it requires high temperature. In southern Karnataka, you see the one more important strain, that is virus and cholera virus is there, but yeah, only the virus DNA. Here for the disease multiplication, is the best season for this the vector multiplication. Usually it's dry or better. So with a low humidity, only in this situation we can say this the problem. Then they are having the different woes for the disease: tobacco, Sanya, petunia, waste, etc. So you see the Gemini viruses. It is a bipartite viruses. It is having single DNA. This is the picture which shows this Gemini particles having DNA A and DNA. It is a begamoviruses. Many we can go the 2% trisodium phosphate for about 25 minutes. Then we can also go for the mulching, silver or black polythene mulching, which is going to repel the white flies as well as the thrips. Then we can go for seed treatment mainly with the imidacloprid at the rate of 5 gram per kg of seeds. So imidacloprid in the market is available in the name of Gouch. So we can use this chemical. Then we can also use of some systemic insecticide like dimethoate, pyridon, acephate. These are very good systemic insecticides for managing this white flies. Then for exclusively for yellow sticky traps, which is very much the important for trapping the white flies. Then use of biocontrol, the fungal biocontrol agent for managing the sucking pest. We can use this Bavarian Lactobacillium as very good agent for managing the sucking pest. Then use of some resistant varieties like Avinash, Mrutyunjaya, Arcanania, Vaibhav varieties against tomato leaf curl virus disease. To respect the viral disease, mosaic virus disease. No, tomato mosaic virus disease, light dog green mosaic model with a dog green area. Is the main point of the disease. You can see the stunted growth. Then the most important one is the leaf. It is looks like a fan like or tendril like. The two things like appearance is the most the characteristic symptom of disease, tomato mosaic disease. Then apart from this, we can see the leaf molding. Then we can see the presence of longitudinal streaks. The presence of longitudinal dog necrotic streaks, mainly on the stem and the root, is also one of the important symptoms. Where the root, we can see the necrotic lesions. Necrotic lesions, mainly on the roots. And we can see the necrosis of the roots. Necrosis of the fruits, also one of the symptom of the disease. This is a disease, and the presence of seeds. But it is the surface-borne disease, but it is not surviving. The second problem is it is spread by leaf contact to data. So management of the host. Then we can go for crop rotation. Okay. Then use of DCT seeds. So the seed is for 15 to 20 minutes. So for managing this, then